Hi everyone, in this demo we're going to explore the Feature Explorer. We're going to start with searching for the human BRCA2 gene, and then we're going to look at the transcripts of BRCA2, look at the exon structure, a few external references, and then also a bit of homology relationships that you can find for the BRCA2 gene. To start with, we're on the Ensembl homepage, and from here I'm going to click on Genome Selector. I can either click on this icon over here, or I can also click on Genome Selector icon just at the top on the navigation bar. Once you're on the Genome Selector page, you're going to see most popular species. We're going to search for human. We've got 565 results, and from here I'm going to select the first one, which is human, GRCH38, release 2026, integrated. Let's add this. And from here, I'm now going to search for a feature. There are different ways that you can search for a gene in Ensembl. We can either use this search option here to find a feature in the selected genome, which is human, or you can also click on this lozenge over here and then look for a gene. I'm going to type in BRCA2, and then I'm going to click on Find. We've got our search results over here, and you've previously looked at Genome Browser. However, this time we're going to explore Feature Explorer. So I'm going to click on Feature Explorer, and this takes us to the gene page for BRCA2 gene, the human BRCA2 gene. At the top navigation bar, we've got the gene name, the Ensembl ID for this gene, the biotype, the strand orientation, and also the coordinates. The coordinates are formatted as the chromosome number, and then after the colon, we have the start position, hyphen, the end position. We've got the main view over here. We've got our different tabs, the transcript tab, gene function that gives you a little bit of protein information, and gene relationships, which gives you homology information, and we're going to look at it in a bit. If I scroll down, we've got our main select transcript. So main is matched annotation between NCBI and Ensembl API. You can click on more information. This also gives you the match from RefSeq and CCDS. And if you would like to download the sequence, you can download the transcript sequence, and you can also download the gene sequence. And then we've got the rest of the BRCA2 transcripts underneath. Before we move on to the different transcripts, I'd like to bring your attention to the right-hand side of the page. So on the right-hand side, you can open or close this panel by clicking on this arrow in the action bar. We've got the overview, so BRCA2, and we've got the Ensembl ID, the gene name. You can also link out to HGNC from here, and we also have publications associated with BRCA2 in this EuroPMC link. Then we have some external references associated with this gene, and here you can find gene external references from Expression Atlas, gene cards, MEM, and then also for the different transcripts. So for the different transcripts, you can click on the individual transcript, and this is then going to give you the CCDS match and also information from Human Protein Atlas, PDB, Reactome, RefSeq, and also UniProt. We're going to come back to the main section of the Feature Explorer, and if you would like to find out further information about a transcript, so for example, if we focus on this main select transcript, you can click on the transcript ID, and then you'll get the option to either open this in the Genome Browser app or the Feature Explorer. We're going to stick with Feature Explorer, so I'm going to click on this icon over here, and this is now going to take me to the Transcript tab. We've got the transcript ID, the biotype, and also the orientation and location. On the transcript tab, you can find the exons tab, which is going to give you the exon table. So we've got the exon ranking number in the first column, and also the exon IDs, the start and end position, and also the length and the sequence for the exon. You can also collapse the introns if you would only like to see exons, and then you can also obviously download all of this data. Furthermore, we also have transcript function, and over here we've got some protein information for this main select transcript from UniProt, InterPro and PDB. We're going to go back to the Gene tab, and to do that we can just go to the right hand side of the page onto the right hand side panel, and underneath the gene name we're now going to click on the Feature Explorer app again, and this brings us back to the gene page. We're now going to look at gene relationships, which gives us a bit of homology information. So we've got some reference species over here. As you can see, we've got chimp, we've got macaque, mouse, quite a few others. You can find their common name, scientific name, and then also their protein similarity and their corresponding gene IDs in other species. An ensemble is not just human. You can find many other species, from agricultural animals to non-vertebrate species like plants, protists, and metazoan species. From this table, you can also see that we've got homology from blue whale, and the protein similarity is 71%. And you can find out more information about the blue whale BRCA2 gene by clicking on the gene ID for it. And now we're on the gene page for blue whale BRCA2. To go back to the home page, you can simply click on the Ensembl logo.